A very good afternoon to everyone. Today we will study about biomolecules. The name itself signifies that biomolecules are molecules that are present in the biologically active compound. Basically, biomolecules are carbon-based organic compound and it is produced by living organism. About 25 chemical elements are present in a living body and all these are made up of carbon, nitrogen, oxygen, hydrogen, phosphorus and sulfur. These biomolecules are basically categorized into two parts. The first one is microbiomolecule. Microbiomolecules are the molecules that are micro in size and they have a low molecular weight, less than 1000 kilodalton, and they are smaller in size. It includes sugar, fatty acid, amino acid, neurotransmitters, nucleotide, vitamins and hormones. But these are the basic building blocks of macromolecules and of a living organism. The macromolecules are the polymer of these microbiomolecules and these include protein, carbohydrate and nucleic acid, which are the basic building block of our body or structure and the genetic material. The major family of small biomolecules or microbiomolecules include sugar, fatty acid, amino acid and nucleotide, and all these four molecules have covalent bonds. From four basic small biomolecules or microbiomolecules, first we will study about amino acid. Amino acids are made up of carbon, hydrogen, nitrogen and oxygen, CHON. These are the monomer of proteins, and when different amino acids are bounded together, they form a stretch of protein that is the building block of our muscles and other structures. These amino acids are called as amino acid because they have two groups together, that is, first is the amino group and the second is carboxyl group. When two groups are bounded together, CO- and NH3 are together filling the valency of a carbon, then that carbon is called as the alpha carbon. The third valency is filled by hydrogen and fourth is by the variable chain or a hydrogen. When at a place of this R variable group hydrogen is present, then it is called as glycine, that is the simplest amino acid. When it is replaced by other CH3, CH2 molecule or other carbon and hydrogen molecules or the methyl groups, then they make different types of amino acid. Thus, an amino acid has this structure where there is a alpha carbon present having amino group, carboxyl group, hydrogen and a variable chain, making different types of amino acid, and then these monomeric amino acid further form the basic structure of a protein. From all the amino acids, we know the most important and the one to be remembered is proline. Proline is basically an amino acid and it is a secondary amine having a secondary amine group, that is NH. A proline molecule has a alpha carbon and a CH2 molecule, and then the two valencies of nitrogen is fulfilled or satisfied by one alpha carbon and second by CH2 group, forming a cyclic ring or a secondary amine. So proline is very important to be remembered because it has a different structure than the other amino acid. Further, the amino acids are divided into different types: beta, gamma, delta and epsilon. That depends on the carbon molecule or the atom having the NH2 group. Means if we have a stretch of amino acid, this is the alpha carbon, this is the beta, this is the gamma, this is the delta and epsilon. If this alpha carbon is bounded with the COH group and having this, and it is having a NH2 group, then it is called as the simple amino acid or the alpha amino acid. If this NH2 group is present at the beta position, then it is called as beta amino acid. If this NH2 group is present at the gamma position, then it is called as the gamma amino acid. If this NH2 group is present at the delta position, then it is called as the delta amino acid. Or if this NH2 group is present at the epsilon position or the epsilon carbon, then this is called as the epsilon amino acid. So on the basis of the presence of the amino acid on alpha, beta, gamma, delta or epsilon carbon, the different types of amino acids are named. Now if we keep an amino acid in 3 different solutions at low pH, acidic condition, intermediate pH 7.4, or the neutral condition, or in a basic condition or high pH, or that is approximately 14, then amino acid in that solvent shows positive charge or neutral charge or zero charge or a negative charge over it. In a low pH or the acidic condition, the positive charge is generated on an amino acid, and that is because of the protonated NH2 group, that is NH3 plus. But at intermediate pH or the neutral pH, there is no charge on an amino acid, and that is because of the neutralization of the negative charge of the carboxyl group and the positive charge of the NH3 group. While at basic pH or the high pH, an amino acid shows negative charge, and that is because of the protonation by a carboxyl group. Thus, in 3 different solutions or solvent, when we put an amino acid, it shows 3 types of charges: positive, neutral or negative charges, and it shows its solubility. Now coming to the optical properties of amino acid. The simplest amino acid is glycine, and it is not an optically active compound. But what you mean by an optically active compound? The optically active compound or an amino acid that is optically active is an amino acid is the molecule when focused by a plane polarized light from a laser beam can rotate the plane of polarized light into clockwise direction or the right direction, or in anti-clockwise direction or the left direction, is called as the chiral molecule or optically active compound. The property of that molecule is called as the chirality. But the rotation of the plane of polarized light can be in the clockwise direction and it can be anti-clockwise direction. But the property consisted by that molecule is called as the chirality, and the molecule is called as chiral molecule. All of the amino acids are chiral in nature and they have a chirality except glycine. On the basis of rotation of plane polarized light, amino acid can be of 2 types. The first is dextrorotatory, where the plane of polarized light is rotated clockwise and it is given a positive sign. The second is levorotatory, where the molecule or the amino acid rotates the plane of polarized light anti-clockwise and it is given a negative sign called as levorotatory. Now, what is achiral molecule? The molecule or the amino acid not showing the chirality or the chiral property called as the achiral molecule include the glycine. The achiral molecule cannot rotate the plane of polarized light either in the right or the left. The optical property of a molecule is measured by an instrument that is called polarimeter. The optical activity of a molecule always have a quantitative value, magnitude and the sign of direction in which it is rotating the plane of polarized light. The value of the rotation of plane of polarized light by a chiral molecule depends on concentration of the solution in which it is kept, the length of the path that is covered by the polarized light, the temperature at which the experiment is performed, and the wavelength of the light that is used here. So the formula of calculation of optical rotation of a compound or a molecule is this, where the optical rotation is calculated in degree, and it is calculated by a specific rotation of the compound at a specific temperature in degree Celsius, and wavelength in centimeter multiplied by concentration of the solution in which it is kept in gram per milliliter multiplied by length of the path covered by the plane polarized light in decimeter, and when these all together multiplied, then this gives the optical rotation of that molecule. A question arises, what are standard amino acid and what are non-standard amino acid? In a living organism, about 100 amino acids are present, but only 22 amino acids are bounded to the ribosomes and then they form protein. These type of amino acids are called as standard amino acids, and these all standard amino acids are L-alpha amino acid, means levorotatory alpha amino acids. The whole standard amino acid, 22 amino acids, can be categorized into two groups. Amino acid having the side chain, if shows non-polar nature, means not showing the polarity, then it includes the amino acid glycine, alanine, valine, leucine, isoleucine, proline, methionine, phenylalanine and tryptophan. If that side chain of that amino acid is showing polarity, then that amino acid, if kept at neutral pH and further shows uncharged or behave as an uncharged molecule or doesn't have any charge, then it includes amino acid serine, threonine, cysteine, asparagine, glutamine and tyrosine. But the amino acid, if shows charge positive or negative at neutral pH having a polar side chain, then it can be further categorized into two subgroups. If it is showing the negative charge, then it includes amino acid aspartate and glutamate, and if that amino acid is showing the positive charge, then it includes lysine, arginine and histamine. Thus whole 22 amino acid that are standard can be categorized into groups and further into subgroups based on the polarity of their side chain and the charge or they show when they are kept in a neutral pH. Now, what is selenocysteine and pyrrolysine? Selenocysteine is 21st amino acid and it is represented by SCC or U, and it has a structure similar to cysteine amino acid. It has selenium rather than sulfur that is present in the cysteine. This is coded by a codon that is UGA, that is a stop codon. This selenocysteine is the modified amino acid of the cysteine amino acid. Second is the pyrrolysine, that is the 22nd amino acid, and it has structure similar to lysine amino acid and it is represented by PYL or O. It is present in the bacterial protein and the codon that forms pyrrolysine is UAG.